Hi, I'm Monse Murel, and I'm the Director of Applied Research and Innovation at Syntego. In this last session of the day, we will talk about how CRISPR is continually evolving through new innovations. Leading genome engineers will cover state-of-the-art on new CRISPR-based technologies, how they are improving the actual methods and leading to new applications. It's a pleasure for me to introducing the chair of this session, is Jared Carlson-Stevermer, the Associate Director of Discovery in the R&D Department at Syntego. Jared received his Ph.D. in biomedical engineering from University of Wisconsin-Madison, where he focused on creating new CRISPR tools to correct complex genomic mutations. At Syntego, he works to develop new techniques and methods for high-throughput genome engineering and implemented them in an automated fashion. Jared, it's a pleasure for us to having you here.
Awesome. Thank you so much, Monse, for the introduction. I just want to welcome everybody to the third and final session of the 2021 World CRISPR Day. Still lots in store for the rest of the day, including the Women of Crispr panel and also networking and entertainment. Without further ado, our first speaker is Lucas Harrington. Lucas is the co-founder and CSO of Mammoth Biosciences, where they focus on how to leverage novel CRISPR systems to provide permanent cures for genetic diseases, and also in the theme of the day, democratizing CRISPR technologies for disease detection. We'll be talking to us today about using compact CRISPR proteins for genome engineering. Lucas?
Thanks, Jared. Let me share my slides. Can everyone see okay? Yep. All right, cool. So I've been told that Jared's going to keep a tight ship in terms of being on time, so I'll try and be efficient. But I'm talking to you today from Mammoth Biosciences, where, like Jared mentioned, we're working on really fundamentally trying to take novel CRISPR discoveries and turn them into products. And so I'm going to talk kind of on two different stories there today. Mammoth was founded about 4 years ago out of Jennifer Doudna's lab, and a lot of the foundational work was done there that we're now working on commercializing. This being the innovation session, I'll really try and start with kind of what that foundational discovery was, and then work our way into what the applications are. And there's two areas that we're really pretty focused on at Mammoth in terms of this area. And so one is therapeutics. And here I'll talk about compact CRISPR proteins and how we've leveraged the diversity of nature to actually cut the size of CRISPR proteins that people are using by about a third. And then the other is diagnostics, where we've been working, especially with COVID over the last year and a half, to bring decentralized diagnostics to a broad array of people. And so I think if there's really one thing that I want people to take away from today's presentation, it's really that Cas9 and Cas12a, if you've used those systems, are really just the tip of the iceberg in terms of the capabilities that CRISPR has to offer. Without question, Cas9's been an extremely powerful tool, and a lot of the results that have been coming out of the clinic this year really highlight that. But there's tens of thousands of different CRISPR systems out there. And to think that just this one size fits all, this single tool is the one that we should be using for all the applications, I think it leaves a lot on the table for other applications. And so at Mammoth, really what we're focused on is taking those tens of thousands or maybe even hundreds of thousands of different flavors of CRISPR that are out there and trying to understand what's the fundamental biology behind them. And then trying to actually leverage that diversity to push CRISPR into areas that it's not able to be used for right now. And like a lot of groups, this starts with metagenomic data. SpyCas9 was really the first system to be used, and that's now nearly a decade ago. And the amount of the genomic universe that's been sequenced since then really has exploded. And also the tools that we use to actually mine that diversity and find new CRISPR systems has similarly increased. And so that's really where a lot of the opportunity is looking at these uncultivated microbes and trying to understand how they protect themselves against bacteriophage and pulling those tools out of those metagenomic samples. So at Mammoth, we've set up a pipeline to do this, and there's been a lot of work to automate this process. But it starts with actually sourcing that metagenomic data. There's a lot in public data sets. There's also maybe even more held in private data sets that you can access through proprietary agreements that you might have. From there, we drive it into a bioinformatics process where we use both homology and non-homology based methods to try and either expand the diversity of known CRISPR systems or define wholly new systems that we didn't know about previously. And really where the work comes in after that, not to undermine the effort of the bioinformatics teams, but it's really in trying to take those just massive number of sequences that are in the computer and trying to understand which ones are the actual good ones, which are the needle in the haystack that are going to be useful for genome editing or for diagnostics. And so we've set up this process that's a very high throughput, automated. It's just a string of liquid handlers essentially that go and characterize the CRISPR systems and identify what are its functional components, what are the features of the system, and then allow us to actually try and use that to understand what applications they're going to be good for. And so here I've listed out a few of the systems that we work with at Mammoth, Cas14 and Cas-Phi that we'll talk about today, new variants of type 5 systems, new variants of Cas13s, so type 6 systems that we use for RNA targeting. And really what comes out of this pipeline is a menu of proteins. So instead of just having your Cas9 or your Cas12a, you can actually go look at the repertoire of proteins that you've built up and try and pick the best protein for whatever the particular application is. So here's some listed over here on the right. And we get asked a lot, so mining that diversity is great, but there's also a lot of groups that you'll hear from today as well that are doing, engineering. And we really at Mammoth see those as two complementary skill sets that really should be combined to have the best CRISPR systems possible. And so discovery is really about, actually exploring that diversity that we described on the previous slides. And here you're really making evolutionary leaps where you're looking at systems that might have less than 10% of the identity between the different CRISPR systems in terms of the protein sequence. So that's really where you find completely new properties that you wouldn't expect. And then after that, at Mammoth, we take that and feed it into our engineering pipeline, which is really about trying to expand, in smaller ways. So maybe mutating a handful of amino acids in the protein to actually enhance a particular property or find an enzyme that's going to be useful for something that you can't find in nature because, evolution hasn't actually driven it down that pathway. So I think this is a really great session today because you really have people that are representing kind of both sides of these coins. And I think it'll be very interesting to just see that comparison. So now I'm going to dive into two very quick examples of how the diversity of CRISPR systems has opened up new areas, in particular areas that we're pushing on at Mammoth. So the first one is in therapeutics, and that's really about using compact proteins for gene editing. And so, like I said, I'll start with the fundamental discovery and then talk about the applications that it enables. So with the fundamental discovery, and this going back quite a few years, back to about 2016 or 2017, we started to look at different versions of CRISPR systems and started to see systems that were much, much smaller than what we'd expect to see. So Cas9, for reference, is the typical Cas9 is almost 1,400 amino acids. We were seeing systems that were about a third of the size of that, ranging from about 400 to about 500 amino acids. And this was really a confusing thing. What were these systems doing? How were they functioning and having the same activity? And so, we went through the process in a much more low throughput way. This is back in the Doudna lab to try and understand how these systems work and what they need. And skipping over quite a bit of work to optimize and develop the systems, the teams have really actually pushed these systems to be on par with those much larger CRISPR systems like Cas9. So here what we're showing is just an assortment of data from primary T cells, obviously a very small subset of data for what's been done with these new CRISPR systems. But here on the left, we're just using mRNA to target these T cells, and here targeting B2M. And you can see just in the head-to-head, we're getting between 80 and 90% targeting with both Cas-Phi and Cas9. And then in the middle, we're actually doing RNP delivery. So preforming the complexes and then delivering them via nucleofuction to the T cells. And here we're actually targeting multiple sites simultaneously. So it's much more of a pressure test to see how well the systems are working versus when you're targeting just a single locus. And here, these are pretty typical targets for T cells, so TRAC-B2M and PDCD1. And what you can see is that similar to the mRNA delivery, we get about 95% for a single target. And even more exciting is when we combine all those systems, all those different guides together and deliver them to the same cell, we're still able to get about 76% knockout of 3 genes simultaneously. So this was, pretty surprising to us that these compact systems can perform very similarly to the much larger components of like Cas9. And I think this kind of raised the question of, and we get asked this a lot, is just how can a system like that do the same activity without having quite as much, muscle, just as much mass as Cas9 does. And a structure of these systems, these Cas14 systems that we were describing on the previous slides, was published about a year ago now by Narecki's group. And what you can see is that the systems actually form a homodimer. So 2 different Cas14 monomers come and scaffold onto a guide RNA to make a system that's much more reminiscent of what we think of as a CRISPR system in terms of the size and just the overall architecture of the protein. So really remarkable example of where evolution's come to a solution that if you were just doing protein engineering, it'd be very hard to land on this solution where you have two monomers coming together. In terms of the applications, why would you care if a system is smaller? This is pretty critical when you're thinking about viral delivery. A lot of the approaches with Cas9, you might use 2 AAVs to deliver something to an organ, which obviously has the inefficiency of needing those to go and actually fuse together and target the same cell. And what we're able to do with these compact systems, they really only fill up about half of the capacity of a typical AAV. Whereas if you tried to use SP-Cas9, you could fit it in, or you couldn't even fit it in. If you use SA-Cas9, you can really just fit in the protein. And so leaving this other half of the AAV open, you can start to think of much more complex reactions that you can do inside the cell, where you're doing, all-in-one HDR and encoding the donor and the CRISPR system into the same AAV, doing multiplexing, a lot of the new modalities that people have been working on, like prime editing, base editing, and CRISPR-A, CRISPR-I, all require other proteins. And you now have the ability to actually put that into one viral vector and have the efficiency and, from a commercial perspective, the manufacturability of just dealing with one AAV. And so size is just really one of the examples of the things that nature provides in terms of the diversity of CRISPR systems. But really, it's quite a menu, and anything you can kind of dream up that evolution could be engineering CRISPR systems for. We generally believe that evolution's probably done that over the course of the hundreds of millions of years of time that it's been able to hone these systems. So that's compact CRISPR systems. I'll now jump into diagnostics, so a very big gear shift. And this is really one of the big things that we've been focused on for the last year is how to use CRISPR, not just for gene editing, but for diagnostics as well, especially in response to COVID. And so again, I'll start with the foundational discovery. And here, this was a very puzzling finding that we had now about 3 years ago, where what we found was that type 5 systems, originally this was Cas12a that we were testing it with, what we found is that in addition to going and cutting their cis targets, so going and cutting and making a double-stranded break like we're all familiar with, these systems also, once they're activated, once they're bound to a target, can then go and catalyze non-specific single-stranded DNA cleavage. So that's what's shown here, where we have a Cas12a that's bound to an activator, and we're incubating it with this M13, so single-stranded DNA bacteriophage. And what you can see is that there's rapid degradation of the starting fragment down to very small pieces. In contrast with SPI-Cas9, you don't see such an activity, and it just sits there and doesn't really touch this. So this was very puzzling, but immediately we started to think about how this could be leveraged as a tool. And what came to mind was actually using this for diagnostics and actually leveraging that single-stranded DNA cleavage activity to detect a nucleic acid sequence. So this is how the platform works, which is called Detector. Basically, you have a CRISPR system that you've programmed to go find a target. You incubate it with a short oligonucleotide that has a fluorophore and a quencher. There's other ways to do the readout, but this was the first way that we approached it. And what you have is that once that CRISPR system goes and finds its target, it then goes and cleaves that fluorophore and quencher pair and releases the fluorophore so that it can give it a real-time signal that it's found that sequence. And so, of course, we were focused on quite a few different targets, but at the beginning of 2020, we were collaborating with someone named Charles Qi over at UCSF, who is just beginning to hear about this new pandemic that was arising that we're obviously all familiar with now. And he, really alerted us to us and said, even when there were just a couple of cases in the United States, and said, this is going to be something serious. You definitely should get on this and start working on it. And so he had some of the first patient samples at the time. I think the outbreak was up in the Northwest. Again, just a few cases in the entire United States. And the team really began to focus on this as a target to go and be able to help actually, alert people when they have COVID and stop the spread of COVID-19. And I won't go through all the different things that have happened over the last year, but I think the real message here is just how versatile and agile this platform is. To be able to take a new technology, so CRISPR Diagnostics, and have a really robust assay that can be used on clinical samples in such a short period of time is really remarkable. And it's really the same feature that makes CRISPR such a powerful gene editing tool, is that even someone with a high school education can go and design a new guide to target a new site in the genome. And the same principle holds here, where a new variant arises or another pandemic comes about, and you can simply swap out the components that are matching the target to then have a new diagnostic assay. So that's really what we've been focused on with COVID-19, is helping to help support and build up the infrastructure for centralized testing, and really supporting the efforts to increase the capacity, have our orthogonal supply chains to have testing be much more robust. Long term, what CRISPR enables is really to move diagnostics out of that centralized capacity and put it much, much closer to the patient and give results in a time frame that's much more actionable and really makes us think about the value of diagnostics in a completely different way. And so here's a mock-up of the prototypes that the teams are working on, but allowing you to have something eventually in your home where you can actually find out whether you're infected with a certain thing, whether you have a particular SNP that makes you predispositioned for something, but really changing it from this physician-controlled data to something that the consumer can then go and interact in an intelligent way with their data. So that brings to a close the two different vignettes that I wanted to chat through of how fundamental CRISPR biology can actually lead to really big applications and new applications in terms of how we wouldn't have thought to use CRISPR originally. And all this has been supported by, of course, the Mammoth team, who's now about 115 people in the company. And it's just such an honor to be able to work with all these people. And a big thank you to them. And with that, I think I'm on time, Jared. So hopefully I don't get reprimanded.
Awesome. Yeah, perfectly on time. Thanks, Lucas. Absolutely great talk. I know when we first saw the Cas12a and the diagnostics that you're doing, we just thought that this was an absolute game changer for the entire field, really. We have a bunch of really great questions from the audience. So just first from Sheena, what's the off-target profile of these newer proteins compared to well-characterized Cas9 enzymes?
Yeah, so it's pretty diverse. I think that's another one of the features that you can actually go and mine and choose the right tool for the right job, right? In some diagnostic assays, you actually want something that's going to kind of open up the window and maybe look at different variants of something in one target. In general, a lot of these type 5 systems do seem like they are higher fidelity, and this is true even for Cas12a than Cas9. There's kind of a fundamental difference between how they recognize their target sequences and allosteric regulation of the different domains. But yeah, lots of details in that. But in general, I just say it's diverse, and it's one of those things that you can pick the right tool for the right job, and find things that are higher fidelity for gene editing, or that are more promiscuous if you want to capture more sequences.
Yeah, it's amazing the kind of diversity that evolution has put out there. Along those lines, a question from Tatiana: is there any information about effectiveness and, I guess, off-target activity of new compact Cas proteins when using them in vivo?
Yeah, so we haven't published our in vivo results with those compact systems. Generally, yeah, again, we see them that they're on par or better than Cas9 in most of those settings. I think always you have to think about off targets in a specific context, right? I think it's a challenging thing to just take things out of literature and compare apples to apples. You really have to look at the dose and what's the on-target efficiency that's happening versus just kind of looking at the raw numbers for off targets. Yeah.
Yeah, absolutely. And just one final question from Jimmy for right now. Beyond the public and private databases, does Mammoth have a discovery program to sequence uncultured bacteria or phage in nature to identify novel CRISPR proteins?
Yeah, so the team's been doing quite a bit of work on that. And I think one of the big things that's been interesting in terms of bringing it closer to Mammoth in terms of doing those sequencing studies is actually being able to get a lot more than just the genomic DNA. So as either we go out or have other people go collect samples, you can go then sequence the RNA. You can get what's called metatranscriptomics. You can also look at the bacteriophage, and you can almost start to do experiments in silico where you're seeing what the system targets and what the RNA species are that it's using without actually taking it out and doing it in the lab. Yeah.
Yeah, fantastic. As I mentioned, there's a bunch of really, really great questions that populate from the audience. So maybe we can just take a couple of those offline and get answers to this really, really exciting topic.
Perfect. Yeah. Thanks, Jared. And thanks everyone for listening.
Thanks so much, Lucas. I'm super excited to introduce Jonathan Gutenberg and Omar Abudayyeh. Omar, I will get that correct one of these days, I promise. But Jonathan and Omar are McGovern Institute Fellows at MIT Brain and Cognitive Sciences Group, where they run a joint lab focused on using biological insights to engineer new molecular tools. Welcome, you guys. Super excited to hear about your new technologies that you're developing.
All right. Yeah. Thanks for having us. So we're going to share my screen, and I'm going to give the first half on sort of our work on diagnostics, and Jonathan will dive in for the therapeutics side. Great. Yeah, so I'm just going to hop in. Our group at MIT basically, focuses on making better molecular tools for gene editing and cellular engineering. And as many of you, are probably familiar with, we, mine natural diversity for new, systems, enzymes that we can use to leverage for these applications. And, when we first started down this path now, almost, 6-7 years ago, of course, Cas9 was really the only slice that folks were focusing on for programmable sort of double-stranded DNA cleavage and editing. And we asked the question of whether we could go beyond Cas9, as other folks in this session have also been exploring. Now, a lot of the work we do relies on finding signatures of systems that we would want to find interesting. So for CRISPR systems, there are a few things that stand out. One is the CRISPR array. The other are proteins that are highly conserved, like adaptation proteins. And we can use those as a seed to mine basically hundreds of thousands of genomes and metagenomes to really find new proteins, which we cluster and can build new families around. And when we did this originally, we found, of course, the type 5 and type 6 CRISPR systems, CPF1 and C2C2, or Cas13a, now it's called, were kind of the first examples of that. And as us and others on this sort of conference have shown, these are really diverse classes, right? Type 5 systems are, I can't even keep track of the lettering that it's gone down to. I think it's like Cas12K and L. I think someone showed L most recently. So now everyone's trying to find Cas12M. But and type 6 now is down to, I think E, Cas13E. So we've really learned that there's such a remarkable diversity of proteins and RNAs, and as fungus published most recently with ancestral systems, there's probably hundreds of thousands or millions of RNA-guided nucleases that exist out there. So it's really remarkable, kind of the Cambrian explosion of our understanding has been for CRISPR diversity. Now, to dive into our diagnostic side of things, a lot of that has been around Cas13 and Cas12, and it revolves around a very interesting activity called the collateral activity. And why Cas13 is unique is because while it can target RNA and RNA genomes of phages and viruses, as shown here, and can cleave, we actually found that not only did it just cleave specifically, but it could actually cleave all over the RNA, degrade the RNA, would cleave other RNAs in a cell, and eventually could lead to even cell arrest and cell death. We first found that in the tube because we were incubating basically these beacons, which have a fluorescent label. And we found that, of course, if you incubate the guide RNA without and Cas13 without a target, nothing happens. But if you then incubate a target RNA, we found not only did you see cleavage of this black target, but you also got cleavage of these labeled RNAs. And of course, we were running these out on gels, and we realized at the time, well, hey, these beacons are kind of telling you that there is presence of this target. And so it kind of hints at the fact that perhaps you could use collateral cleavage as kind of an amplification of signal for diagnostics. And that was kind of the aha moment that led us down the path to try to do that. And so instead of using, beacons that you run out on a gel, we were interested in something a little bit more scalable and easy to use. And so we switched to quenched beacons, which are a fluorophore quencher on a probe. And when they're cleaved, the fluorophore is allowed to fluoresce. And we basically showed that you could use this to target Cas13s against specific transcripts, and that was diagnostically relevant. Now, of course, these systems are not that sensitive. They go down to maybe 1 million copies or hundreds of thousands of copies per microliter. To build out later sensitivity, we had to combine it with preamplification. And so we can take DNA or RNA and put that into like RPA or LAMP or all the 3-4 letter acronym soups of amplifications that exist, as NASBA, SDA, HDA, so on. And that can then be used to transcribe RNA, which then is detected by Cas13. And there you get really good single molecule sensitivity, the stuff that, people can actually use in the clinic or for other applications. So, of course, I don't want to dive into everything we've done in diagnostics world. We've shown infectious disease, bacterial, antibiotic resistance detection, freeze drying and, paper-based testing, genotyping, cell-free cancer detection. I will dive into some of the more recent work from the past year doing COVID detection. And I think it's kind of been a remarkable kind of test of CRISPR diagnostics, multiple groups. This is, I think, when Sherlock Biosciences, a company we co-founded, first got FDA approval for their lab-based CRISPR test. And, of course, you just heard from Lucas, and Mammoth has their own version of a CRISPR-based test. And, these are pretty cool because it's the first sort of FDA, clinical use cases for CRISPR, kind of beat the therapeutics out there into the market. But, at the time this was happening, we were excited, but we also wanted to go further. Lab-based testing was not the end-all be-all. Of course, there's a lot of other things that can do that. But what hasn't happened, of course, is, this future of like, what's the next smart home, right? Like, it's cool to have like these devices like Google Home where you can like have it tell you the news and weather every morning and like dim your lights to like 1 million different colors. But wouldn't it be cool to have like a Google Home that could also like, take a saliva sample and then tell you if you have like the cold versus flu or, COVID or, any other, thing that wakes you up in the middle of the night and you're like, wow, am I sick? Like, it's 2021 and we still use a thermometer basically to diagnose everything at home. There isn't anything too much more than that yet. So, we really are excited about that vision and wanted to make at least a CRISPR-based test that was more amenable to that. Because, of course, the versions that these early lab-based tests came out with are complicated. There are multiple steps. You have the preamplification, and you transfer that to the CRISPR diagnostic test, and you have to purify the stuff beforehand. It's not something someone can do in their kitchen. So we spent basically January of 2020 when we started getting a lot of emails from Asia about like this virus that was taking hold, and we started working on this new chemistry. At one point we were like literally the only lab open in our building when everything shut down because we were working on this. But we were able to actually combine using this amplification called LAMP with thermophilic Cas12 enzymes into a single one-pot reaction where everything worked together happily. And we actually could take a swab from your nose, you dip it in an extraction buffer that pops the viruses open. We had magnetic beads that bind the RNA that comes out, and then we could take off the extraction buffer, bind the magnetic beads, and then resuspend in this what we call stop COVID master mix, which is really just LAMP and these enzymes that work well together. And then we could actually use a lateral flow compatible reporter, use a fluorescent reporter, and do real-time detection. And it was really simple. We even had like pictures in, I think, the paper, I think they made it in the SUP, of like someone using this like kind of like in a kitchen with like a sous vide machine and a water bath, because it runs at a single temperature, and it's pretty simple to use. And we tested like a couple hundred patient samples and showed that, you know, the time to threshold using the real-time detection approach actually matched really well with CT value as measured by a gold standard qPCR test, which is pretty cool. And we got, you know, a total sensitivity with this of about 93%, and specificity was about 98%, as shown here. So it, you know, worked reasonably well. And you know, going from there, you know, I think, you know, we've spent a lot of time, you know, we've shown multiplexing, we've shown that these things can be low cost and they're quantitative. But as I've said, you know, the really big vision here is taking now these simplified chemistries and getting them to work on a device. And, you know, a lot of that involves figuring out how to lyophilize the chemistry, how do you make it room, you know, shelf stable for a long time, and then building a device. And I, you know, hope in the future we'll actually be able to have this. It's, you know, come along quite well, but, you know, a mini device you can hold in your hand that, you know, runs these chemistries, and probably no one will ever know that there's a CRISPR chemistry, you know, inside of it, but that's, you know, what will hopefully be powering it. And it's something that people can use in any setting. And I think that's hopefully what we're, you know, still working towards and trying to build. Yeah, so on that note, you know, this is, you know, what we've been doing in the diagnostic space, and of course there's a lot of cellular-based applications. So I'll pass the baton to Jonathan.
Yeah, great, cool, perfect. Well, yeah, I think that's a great kind of one half the perspective of how we can use RNA targeting tools or new CRISPR tools for diagnostics. But obviously, one of the amazing things we can do with these tools is we can actually have therapeutic mammalian work, and you can probably see the TC hood behind me. So how do we actually apply these RNA targeting tools in mammalian contexts? And it's really great because there is a lot of actual RNA targeting work in mammalian cells. So there's a lot of RNA in the cell, as you're probably well aware. And that allows us to do a lot of different things where we can actually target RNA and manipulate it. Of course, there's mRNAs, there's other RNAs in general. So being able to actually target that RNA and manipulate it allows us to do a ton of different applications. So what can we do with that? So obviously, one of the first natural things we can do is knock down transcripts in mammalian cells. So we can actually target in mammalian cells a transcript like here a reporter transcript where we have a luciferase, a Gaussian luciferase and a cypridinal luciferase. We can target one of them and use the natural RNase activity of this Cas13a enzyme to actually knock down and cut that mRNA. So what we can see is that if we target this, we can measure the ratio of these two luciferases and say, is this active in mammalian cells or not? And luckily it was. So we actually compared the Cas13 family to a kind of gold or state-of-the-art technique at that time, RNAi, which you obviously are familiar with for knocking down RNA. We found that we could get comparable or better knockdown compared to RNAi. And it's also interesting because you can see that we could try both a cytoplasmic and a nuclear functionalization. So this speaks to the fact that, of course, by putting different nuclear localization sequences or nuclear export sequences on these enzymes, we can actually export them to the cytoplasm or put them in the nucleus where they can target things like nuclear long non-coding RNAs. So this is actually a great proof of principle. Obviously, with these CRISPR-based tools or any of these nucleic acid-based tools, the question is, how specific are they and what's the actual effect on the cell? So what we did is we actually did transcriptome-wide sequencing in these HEK cells, and we can compare the effect of a targeting guide and a non-targeting guide to see how true or non-true to each other are these transcriptomes. So if we plot this for something like RNA interference, you might say, how much deviation from the central axis is there that would cause a non-targeting guide to vary from a targeting guide? And that's RNAi. So there's actually a bit of density along that central axis, which is indicative of the fact that there's a lot of off targets. And if you use one of these RNA targeting CRISPR tools like Cas13a and compare, you see, wow, it's much tighter. And those two dots over there, those are these basically the targeted transcript of the Gaussian luciferase. So we can do this with multiple Cas13 family members. As multiple people in these sessions have mentioned, it's an ever-growing family. So both with Cas13a and doing the same thing with Cas13b, we can get very precise knockdown in these cell types. Now, you're probably saying, wait, that entire first half that Omar talked about was based on this fact that you can target RNA and then activate collateral activity. That's kind of discordant. And when we first saw this, we said, well, that is kind of strange, but perhaps in this cell type and other cell types that we might want to test, that small amount of cutting isn't a problem. And that's great to see, but that isn't always true. So as these Cas13 targeting tools have proliferated, people have started to test them in different cell types, including, in this example, U87 glioblastoma cells. And what groups have seen is that in targeting conditions, there can be reductions in cell growth, there can be toxicity, and there can be, this is the actual ribosomal RNA that you can run in a gel and see that's degraded. So loss of RNA integrity over time due to this presumptively collateral activity. So it makes you think, well, one, not everything that works in one cell type may be transferable to another, and not everything that works in one animal may be transferable to another. People have shown that in certain applications in mice that Cas13 works great, but anecdotally people have had challenges in things like zebrafish. So how do we take an enzyme that has a built-in endogenous RNA targeting capacity, but not have it have this collateral activity? And so you can probably already say to yourself, well, why don't you go back into natural diversity to find more instances of RNA targeting enzymes? So that's what we really asked ourselves. Can we go back to that ever-growing tool of public and private databases, as long as some of information of our own, to actually find another tool? So this is actually a great little piece of CRISPR biology. We talked a lot about class 2s, and Lucas talked a little about these class 2 systems, but really, if you think about how we've used these class 2 systems as a source of tools, people have shied away from this, it's comparable, actually, more abundant source of diversity in these class 1 systems, because there's a bunch of different proteins there. And no one wants to have to transfect or nucleofect 6 different proteins. So really the question is, can we find systems in here that maybe are class 1, the type 3 systems are known to target RNA, but maybe they have fewer proteins. Maybe there's something else there. So what we did is we actually partnered up with Eugene Koonin to explore crystal diversity. And what we found is that there are multiple different type 3 systems. There's a class 1, which is just characterized by having multiple different proteins. But there's something interesting here. The type 3E system actually has all of these different proteins fused together into a single effector. So there's actually multiple Cas7 domains, 4 of them, and a Cas11 domain in this one protein. And that's something that we started calling Cas711. Credit to Eugene on the naming. But the question is, does this enzyme have RNA targeting capabilities, and does it have collateral? So we actually put it into a couple different contexts. We don't have time today to talk about all the biochemistry we've done, but we actually showed that, 1, it's a programmable RNA targeting system, and 2, it has no collateral activity. This is what collateral activity looks like on a graph. You can see that we're targeting one transcript, and we have a reporter in here, and we can actually see the fluorescence build up of that collateral cleavage. There's no actual effect here from Cas711, no collateral activity. And we can actually see that in bacterial cells too. When we target a transcript like RFP with Cas13, you actually reduce the growth rate of the bacteria. It's toxic, and it's actually a programmed cell death mechanism. You don't see this Cas711. So this is really great to see. And we said, well, can we take this to mammalian cells? So here's the same assay, a little bit of an updated picture, 2021, but really we're taking 2 luciferases and targeting each one. And what we can do is and show that now that there are multiple different Cas13s, we actually can be on this target comparable or better, in this case with the Cas13 with this guide, to those RNAi. So Cas711 works fantastic. And we can say in these 293 T cells, do we see any growth defect? No, we don't. So this agrees with what we saw in our previous papers. In 293F T cells, we don't get a growth defect from targeting with these Cas13s or with Cas711 or with RNAi. But now if we start looking into these different cell models that have been shown to have toxicity, we can say in U87 cells, well, one, do the systems still work? So here you can see we still get good knockdown with these systems. RNAi, interestingly, does not work. And this is a known fact. It relies on endogenous machinery that these glioblastoma lines don't have. But even more importantly, if we plot the knockdown here, or the percent expression, so lower expression here is more knockdown, versus the cell viability, we can see things that, for example, PSP-Cas13b is getting loss in viability just from expression, but also additionally with knockdown. Similar things with other Cas13s, but with Cas711, we have the highest viability measured here, as well as a large amount of knockdown. So this is a great system for having collateral-free knockdown for targeting RNA in both biological explorations, where you need to be able to do things in certain cell lines, but also as a therapeutic. So obviously, this is only one piece of this larger comprehensive toolbox of RNA binding proteins, the Cas13s and Cas711s, as well as other RNA targeting enzymes that we are thinking about. And these allow us to do so many things that we can now modulate translation and expression. Groups such as actually David Liu published a recent paper using recruitment of RNA modifying enzymes to modulate the epitranscriptome. And groups such as Patrick Su have shown that we can use these to modulate splicing. So it's really an evolving time. We like to think of it as, if you look at the DNA toolbox, where now there's so many things we can do, and it's growing all the time with base editing and prime editing and CRISPR-A and CRISPR-I. RNA is, we like to think of ourselves a little bit as an underdog here, but it's still definitely a growing toolbox that we're really interested in seeing where it can go. And I know that we've talked today a little bit about RNA targeting, but to take a step back, our group is really focused about how we can mine natural diversity to build these tools for RNA and DNA targeting, and essentially assemble a cellular engineering toolbox. So we believe that genome engineering is part of this much broader context of cellular engineering. And so we're thinking about a couple different ways to do this. So our precise RNA modulation, as we just talked about today, and we're working on ways for efficient programmable gene integration without double-stranded breaks using new technologies. So this is obviously a very active area, but can we enzymatically bypass the requirement of relying on endogenous gene repair? And to go a little bit far from CRISPR in general, but how can we actually use RNA-based techniques in engineering of natural enzymes to understand where a cell is and be able to detect that, as well as control where it's going? So we're very excited about this, future of cellular and part of that genome engineering. And of course, CRISPR is a huge part of that for us. So with that, we'd like to thank everybody. So we're the Abudayyeh Gutenberg Lab, and of course we have tissue culture capabilities, but we're at McGovern, and this is actually a little bit outdated pre-COVID picture of us. We're a bit larger now, but we're actively recruiting postdocs. So if any of these things resonate with you, we'd love to chat. And we'd like to thank everybody that made this possible, including the first author on the Cas711 paper, Austin Ozcan, and our collaborators, Eugene Koonin, who is a big part of the work on 711, Feng Zhang, who is really our old mentor, but really integral in Stop COVID, as well as Jim Collins and Party Sabetti, who helped a lot in early Sherlock work. So thank you so much for listening, and we'd love to take any questions.
Yeah, awesome. Thank you both for such a wonderful chat. I think next time, actually, the challenge is going to be, instead of having the BSc in the background, you have to be physically doing an experiment while presenting slides. That's something I'd love to see. So we'd actually love to do some Q&A, but you guys have already answered most of them kind of in chat. It's like you guys have, you got some predictive capabilities there.
I tried to get him to come to the office, but he could, Jonathan didn't want to leave the lab. So
always working. Just one quick question from Anna. Are Cas13 knockdowns reversible? And also, along with that, how long does the knockdown effect last once cells are transfected?
Yeah, I mean, I can start off on that, but Omar, show your color. So they are reversible. If you do actually have an inducible system, you can withdraw. And But that knockdown level is transcript dependent, of course. Some transcripts, and of course, some transcripts are degraded in different ways in different cells, and different expression of transcripts will mean that they'll restore to their levels in different amounts. So it really does depend what you're trying to target. But that is something that we find really inspiring, because the idea that you can take a reversible enzyme and actually be able to gate it using things like small molecules, you can actually allow for repeated knockdown over time. And you can turn something that's as complex as programmable gene targeting and transcript knockdown into just taking a small molecule. So we encourage your systems of interest to try that out. But we've seen a lot of interesting progress with being able to have these as a temporary tool.
Awesome. Yeah, no, I definitely agree. So there's a couple more questions in chat. Maybe you guys have some time, we can just go in and answer those offline as we move on to our next speaker.
Yeah, thanks so much for having us.
Good.
Thank you, guys.
Thanks for having us. Yeah.
So our next speaker is Stanley Qi. Stanley is an assistant professor in the Department of Bioengineering and Chemical and Systems Biology at Stanford University. His work focuses also on expanding the CRISPR toolkit and includes some of the first proof-of-principle experiments demonstrating the potential of CRISPR-I and CRISPR-A. Today he's going to talk to us about a new system that his lab has engineered, miniature CRISPR-Cas for gene editing and regulation.
Thank you for the introduction, and it's really great to join the Second World CRISPR Day and to present our work. And just a quick overview of what we do in the research lab. And our research focus is to think about beyond the gene editing. And what we have done before is to apply synthetic biology and protein engineering to create novel variants of the CRISPR molecules harvested from nature that you heard from the previous talks, and give them new functions that doesn't normally exist in these natural systems. One example was the use of the Cas9, which by silencing the nucleus domains of the molecule, but still use that as an RNA-guided DNA binding system for controllable genome regulation, known as CRISPR-I, CRISPR-A. And the same principle has been also applied to other systems. So the concept is we have a class of programmable molecules, Cas9, 12, 13, combined with guide RNAs, delivered to the cells, and depending on the nature of these molecules, either they bind to DNA or RNA, so we can use them for some functions beyond editing. So for example, in the CRISPR-I and CRISPR-A, one for inhibition, another for activation, we can use this Cas9, Cas12 system to, instead of cutting the DNA, recruit an effector to turn on or turn off the downstream gene regulation. In a similar manner, we can also recruit enzymatic domains to either methylate or acetylate the DNA or chromatin, to change the chromatin status, to rewrite the epigenetic memory. And as another example, we can also recruit the pieces of DNA to modulate how the DNA interact with each other, and also localized differently in the nucleus as a 3D genome control. And also this tool can be repurposed for genome or RNA imaging by fusing them to dyes. And in the last talk that came to the CRISPR Day meeting, I presented how we repurposed a bacteria antiviral system, which is this CRISPR-Cas13, as a human antiviral system to fight SARS-CoV-2. So really, like, for our interest is we like to think beyond the box, outside the box, to how we can use these tools for different purposes. Despite there are many, many exciting and interesting CRISPR applications for research, for screening, for cell engineering, there still remains a key challenge, which is how can we use these tools more effectively, safely, and programmably in vivo, in animals or in the human body. And if you look at the practical limitation of the CRISPR system, it's not very friendly for delivery. Really, like, I'm using the adeno-associated virus, AAV, as an example. So the Cas system naturally come at the big size, shown just for your reference, and most CRISPR systems do not fit into a single AAV vector. And there are only a few examples that have been discovered before, like the Staphylococcus aureus Cas9, 3.2 kb, or a CG Cas9, 3.0 kb, can barely fit into a single AAV, yet the efficiency is not great. And also further expanding into other applications by fusing the Cas with effectors for activation, repression, base editing, prime editing, or epigenome editing, like what we really love to do. If there is no way to solve this problem, it's definitely not good for a single vector. The question we ask is, how can we apply synthetic biology and protein engineering to make this become a possibility? And that's promote us to work on a much smaller, highly compact miniature CRISPR, we termed Cas-mini. Before we engineer Cas-mini, we look at the naturally discovered CRISPRs. And among the Cas9 family, they are very large, generally larger than 1,000 base pair. Among the Cas12 family, there are some interesting subgroups like Cas12F, also called Cas14, and their size is in general smaller than 1.6 kb. So when we compare the structure, to be honest, when we started this project working on engineering the Cas12F, and there was not a structure reported for this system. So we had to computationally predict it. And then by aligning to other Cas9 and Cas12 the structures. Cas9 has 4.2 kb. It's a large protein with many domains. Compared to that, the Cas12F has a smaller structure and also is from a dimer. And that's why even though at 1/3 of the size of the Cas9, because of this dimerization, it can do something that similarly as Cas9. That's why the system is super compact, but still working. But the first question is, okay, you get a system from bacteria. Does that work in mammalian cells? That's not guaranteed, because these systems came from could be a simpler environment due to evolution. They may not have been prepared to work in a much larger genome, like our human genome, which is 1,000 times bigger than a bacterial genome. So the postdoc Xiaoshu worked on this question by engineering a mammalian version and by also mutating the Cas12F into a dead version. So it still binds, but doesn't cut. That's a hope. And to our disappointment, the natural system did not work at all. And when we tried to activate a fluorescent GIP reporter in the human HAC93 cells, we saw 0% cells can be activated, suggesting the system completely failed in mammalian cells. Then we think about synthetic biology. How about engineering molecules? First is the guide RNA. And we look at the secondary structure of the guide RNA, and we did a few changes. One, to eliminate the transcriptional terminators. Another is to engineer at the interface between the CRISPR RNA and the guide RNA, showing this blue box. And that slightly improves the performance. For example, compared to the version that has no effect at all, that we find one design shows 3.6-fold improvement. But while that's good, right? There's a trend, but it's far from being useful. Then we turn to protein engineering. By computational modeling, we choose different amino acids that are closer to the target DNA because we think that closer residues should have an important function for binding. And second is we mutate that to a positive charge residue due to this electron interaction, because DNA is negatively charged, and if it makes the protein more positive charged, we may increase the interaction with the target DNA. To our surprise, that greatly improved the performance of the gene activation on the fluorescent reporter. As you can see, some mutants even showed more than 100-fold improvement for the performance. And there's also a domain that seems to cluster together. And each residue within that domain shows a strong propensity for improvement. We also used repetitive engineering to iteratively engineer the protein with more mutations. So basically, we choose the good mutants from each round of mutation screening, and then we introduce more mutations by combining the beneficial mutants. Hopefully, these mutations will stack together. And that really leads to a gradually improved performance of the mutants, starting with some wild-type systems shown on the left that has almost no activity. And after each round of mutation, we find better and better variants. And as in the round 4, we find almost 1,000-fold improvement over the original system. So we also ask questions. How about that really compared to the original system on endogenous genes. We tested on about many genes, and shown here just 2 examples: the human interferon gamma and also the human beta globin gene. As you can see, the gray bar shows the original system performance, almost nothing, and the color bar shows how much we can activate this gene. And since these axes are on the log fold, so you can see there's a great improvement. And we also defined the requirements for the guide RNA to bind. And not every guide RNA will work. So to summarize this data, we test many genes and many guide RNAs. For each gene, we designed 10, 20 guide RNAs. And we found about 20% of the guide RNAs showed activation. And also, via this process, we better define the PAM sequence to be TTTR. So the next question is, how does this Cas-Mini system seems working well, but how does it compare to the other system? To do that, we compare to Cas12a, which is CPF1. As you can see, Cas-Mini has a much smaller size. We target design guide RNA, and for both systems target to the same region of the GFP reporter, because they can use the same PAM sequence. And the Cas-Mini showed even better activation on this reporter gene. When we compared across many endogenous genes, our conclusion is the Cas-Mini activator either showed similar performance or better performance on these genes, so which is very, very good. And the next question is the specificity, how many off targets? And then when we engineer a CRISPR system to be more effective, there is a natural question that also become less specific. To do that, we performed genome-wide RNA sequencing, and by comparing a targeting guide versus a non-targeting guide, and how does that affect the global-wide transcriptome expression. And the red colors show the Cas-Mini influence on the endogenous genes, and the black dots shows the Cas12. From this data, you can see they are very comparable and not much off targets, or no off targets. And while the on-target gene is strongly activated. So we also use this engineered version to test on gene editing, since the system came as a nuclease system. We test on different genes and different loci and achieved about 10-15% of gene editing efficiency. We also compare different mutants and to see what's the editing window. And because people know Cas9 has a very narrow editing window, ±1 is a dominant editing. And for our system, we find a much broader editing window. More than 20 bits per DNA is deleted. So that would be a good news for knocking out a gene, because that would be a more complete disruption of the target gene. We also fuse the Cas-Mini to the adenosine deaminase, which will mutate a single A to a single G. And we test different protein engineering constructs, and we found some constructs allow us to achieve a decent number of base editing at different genes. And also the editing window is very narrow, which is a good news because it doesn't affect A's outside of the editing window. And as you can see, the editing window is usually the third and fourth base pair. And we think the Cas-Mini system really brings in new opportunities to work with this very limited delivery capacity of the many systems, including adeno-associated virus, and even for mRNA delivery, smaller payloads usually can be delivered more efficiently. And that's particularly pronounced when we work with different functions, repressor, activator, base editing, prime editing, because from our prediction, they all fall below the packaging capacity of the AAV. And so far in this work, we demonstrate the use of synthetic biology to turn a originally non-functional system, Cas12f or Cas14, into a completely working system, and also not only working, but working comparably well with other defined gold standard CRISPRs, and also demonstrate their use for different functions on the genome. As you see that this could be useful, very more friendly for different delivery approaches. With this, I'd like to thank the lab for contributing the work and providing the useful tools to the research community, and also enables potential use for the clinics. And in particularly the leading author, Xiaoshu Su, who's diligently engineered the protein and tolerate all the failures and until the system working well. And also the funding source. Thank you very much.
Awesome. So unfortunately, we don't have any time for questions for Stanley. And we're sorry that some of you are experiencing difficulties seeing the slides to go along with the talk. You will be able to come back and see the completed talks again later on. These are all recorded along with the slides, so you'll be able to get access to them. But in good news, we do have Erin back. So we're going to be to hear to her a little bit more about her work with transcriptome editing using ADARs.
All right. Thank you so much for the patience. I know as scientists, we're all very used to technical difficulties and improvising. This again is talking about my work in Peter Beal's lab at UC Davis, looking at nucleic acid editing using ADAR enzymes. Next slide. We skipped one. There we go. This work focuses on the adenosine deaminase acting on RNA or ADAR family of enzymes, and ADARs deaminate adenosine to inosine and double-stranded RNA. Inosine is recognized by cellular machinery as guanosine, and so this affords an A to G transition. Next slide. ADAR's ability to deaminate adenosine to this guanosine mimic gives us the potential to correct diseases related to G to A mutations, which is significant because the majority of genetic variants associated with disease are these single point mutations, and the most prevalent are G to A. ADAR is uniquely suited to targeting these point mutations because it will only act on duplex RNA, meaning that we can direct it to these disease-causing sequences by delivering a guide strand of RNA that is complementary to the target sequence. The effects of RNA editing are transient, so any off-target edits do not result in a permanent change to the genome. The downside then being that RNA editing therapeutics require continuous administration, but it does give us a reversible and tunable solution. One of the major advantages of using ADARs in this therapeutic context is that the enzyme is already expressed in humans, which eliminates the need for enzyme delivery. Other editing technologies requiring the use of exogenous enzymes can face limitations based on immune stimulation and delivery barriers, but using endogenous ADARs only requires the delivery of guide RNA to elicit their therapeutic effect. However, current methods of site-directed RNA editing by endogenous ADAR enzymes are limited by low efficiency, and this has inspired efforts to optimize guide RNAs that promote editing by endogenous ADARs. So in this project, I've used structural information from ADAR2 RNA complexes to design guide oligonucleotides for site-directed RNA editing. Next slide. The Beal and Fisher labs at UC Davis were able to solve the structure of the catalytic domain of ADAR2 bound to double-stranded RNA in 2016. This structure showed that the enzyme operates through a base flipping mechanism where this edited base is flipped out of the duplex and into the enzyme active site. This leaves behind a base we refer to as the orphan base. And in the preferred editing context for ADAR2, this is an adenosine paired across from a cytidine, so this leaves behind an orphan cytidine. This structure gave insights into the protein RNA contacts that stabilize the flipped conformation of the base, and this includes a contact pictured that's observed at residue 488. And residue 488 makes this suspected hydrogen bonding contact with N3 of the orphan cytidine. In the bottom panel, you'll see the Beal and Fisher labs also solved the structure of a single mutant of ADAR2 at the 488 residue. This is a glutamate to glutamine mutation. And this E488Q mutation was previously discovered by the Bass Lab at the University of Utah because it increases the rate of deamination up to 60-fold for certain substrates. But despite this really large difference in the activity of the wild type and the E488Q enzyme, you can see that their crystal structures look nearly superimposable. Next slide. This consistency in the crystal structures led us to think a little bit more about the hydrogen bond occurring at that position. At physiological pH, this glutamate would be deprotonated, requiring protonation in order to make this contact, but the glutamine residue would be able to donate a hydrogen bond to this orphan cytidine without relying on protonation. The hydrogen bond donation is believed to be the reason for the hyperactivity of that ADAR2 E488Q. And this led to an idea for guide RNA design that if we can make this same protonation independent hydrogen bond with the wild type enzyme, then we could achieve a greater catalytic rate for the endogenously expressed ADARs. But before pursuing this idea, we wanted to further understand the contact made between the wild type enzyme and the orphan base. Next slide. Residue 488 is important in stabilizing the base-flipped conformation of the enzyme RNA complex. So if this contact between wild type ADAR2 and the cytidine orphan base does rely on protonation, then the base flipping ability of ADAR2 should be pH dependent. Next slide. This led us to pursue this assay that measures the extent of base flipping by ADAR2 via fluorescence, and this employs this fluorescent nucleoside analog 2-aminopurine. 2-aminopurine retains its fluorescence in a single strand of RNA, but its fluorescence is quenched when it is hybridized as part of a duplex. Due to ADAR2's base flipping mechanism, it can flip this base out of the duplex and into the enzyme active site, which restores fluorescence. But To explore the pH dependence of base flipping, I measured the fluorescence enhancement of an RNA substrate containing 2-aminopurine with the addition of ADAR2 enzyme. And so the plot you'll see here on the left is the normalized fluorescence enhancement from pH 6 to pH 8.5. And for the wild type enzyme on the left, we see that if pH is lowered, there's a greater extent of base flipping by ADAR2. But this result does not yet allow us to attribute this trend specifically to that 488 residue. So I decided to perform the same assay using the E488Q mutant enzyme, which already has that hydrogen bond donor present. And what we see in the panel on the right is that although base flipping by ADAR2 E488Q was affected by changes in pH, the mutant does not show a preference for either the high or low pH, which is in contrast to the wild type enzyme, which clearly shows a preference for the low pH. Next slide. So this tells us that ADAR2 activity is pH dependent, and that this pH dependence can be attributed to this contact at the 488 residue, which in the wild type enzyme becomes protonated, enabling efficient base flipping and therefore enhanced RNA editing. Next slide. So we were interested in understanding this pH dependent contact because of our desire to design guide RNAs that promote efficient editing via endogenous ADARs. We want to be able to utilize endogenously expressed ADAR enzymes instead of using an enzyme mutation to increase the rate. So therefore, we hope to make a modification to the guide RNA that mimics this contact made by the hyperactive ADAR2 E488Q enzyme. Next slide. To mimic the effect of this mutation, we need to provide a hydrogen bond between residue 488 and N3 of the orphan base that is not dependent upon protonation. And this meant incorporating cytosine analogs with hydrogen bond donors at the N3 position to the orphan position of the guide RNA. And I tested these 3 analogs pictured here, but Benner's base V had the most interesting effects, so I'm just going to talk about that modification. Next slide. First, we examine the effect of this orphan base modification on base flipping, and you will see that I'm using DC and DZ. These are 2' deoxynucleosides in the orphan position, and that's because they're commercially available and they're more metabolically stable. So in our final guide oligonucleotide design, we actually avoid ribose bases altogether. So first in the orange, I looked at the behavior of cytosine in the orphan position, and I saw that by changing from the wild type to the E488Q mutant enzyme, we see an enhancement in base flipping. And using the Z base, I saw that this preference was actually reversed. Instead of seeing an enhancement in base flipping with that E488Q mutation, I saw a decrease, and I observed that this base seemed to enhance base flipping with the wild type enzyme. And so the result here demonstrated that base flipping was enhanced when there's a hydrogen bond donor acceptor pair between residue 488 and N3 of the orphan base. And this is promising since the Z analog paired with wild type ADAR mimics the effect of that 488 mutant. And so we hope that it would also allow ADAR2 wild type to exhibit the same hyperactivity as the mutant. Next slide. And so next I examined how in vitro deamination activity was affected by this orphan base modification. And again, in the orange, starting with the cytosine base and an RNA as a control, I observed an increase in the rate of reaction with this hyperactive ADAR2 E488Q. And again, switching to the Z base in the blue, I see the same trend that I observed with base flipping, that now this base has a preference for wild type ADAR2. But the exciting result is that by changing the orphan base in a guide RNA from having a hydrogen bond acceptor to a hydrogen bond donor at that N3 position, we see an increase in the observed rate of reaction about 3.5-fold for both wild type catalytically active ADAR enzymes. Next slide. So I do want to point out that there are advantages to using texture and chemical modifications as opposed to enzyme mutations that go beyond the use of just endogenous enzymes for editing. So as previously reported, we found that levels of off-target editing at various sites were significantly increased with the use of this hyperactive ADAR2 E488Q, which is what's commonly used in engineered ADAR editing systems. And so you'll see that the levels of off-target editing for this enzyme are in the pink, and off-target editing levels were actually not affected by the use of the Z base in the guide strand, which is comparing both the orange and the blue. However, both the Z base and the ADAR2 E488Q mutation were able to enhance the editing at the target base, but only the Z base was able to do this without also increasing off-target. Next slide. And so this led us to see if we could obtain structural data showing the contact made by the Z base. This work was done by Xander Wilcox in the Fisher Lab at UC Davis, and he crystallized and obtained a 2.5 angstrom resolution structure of an ADAR2 RNA complex containing the Z base at the orphan position. And what this showed us is that the orphan base, Z base, forms a closer contact with residue 488 than orphan cytosine had in previous structures. And so we were able to actually enhance this target interaction. Next slide. So far, all that data is just in vitro, but our goal is to enhance editing via endogenous ADARs, and that leads to this last set of experiments. This was done by our collaborators at Therapeutics with guide oligonucleotides isynthesized, again, containing either cytidine or the Z base in the orphan position. And it is notable that this is a 36 to 38 mer guide RNA design, which is much shorter than the 80 to 150 nucleotide guides that are used by other groups who are recruiting endogenous ADARs. In this experiment, they're transfecting only those guide RNAs, so any editing observed is treated in endogenous ADARs that are already in the cell. And we plotted here percent edited for guide oligonucleotides with either DC or DZ in the orphan position. And we see that this single nucleotide change leads to between a 2.5 and a 3.3-fold increase in editing via endogenous ADARs. And this experiment also demonstrates that the enhancement of endogenous editing via ADARs is not cell type, sequence, or target RNA specific. Next slide. Based on our understanding of this protonation-dependent contact between ADAR and the guide RNA, we used rational designed chemically modified guide oligonucleotides to enhance editing by endogenous ADARs. And these single results showed that just by making a single nucleotide change, we could induce a 3-fold increase in the amount of editing by endogenous ADAR. And so we hope that this will help to inspire further efforts to optimize guide RNA. And next slide. So lastly, a big thank you to Dr. Peter Beal and the rest of the Beal group, also to Dr. Andy Fisher and Xander Wilcox, who did the structural work, Dr. Lenko Van Viet and Sherry Kimmel at ProQR Therapeutics, who did the experiments with endogenous ADAR editing, and to ProQR Therapeutics and the NIH for funding. And lastly, to all of you for your patience and attention. So thank you, and if we have time, I'll take any questions.
Awesome. Thank you so much, Erin, for a wonderful talk. We're so glad we got to bring you back on and know how difficult it can be starting and stopping a talk midstream. And thanks to the audience and for just bearing with us. It's really exciting to hear about advancements in guide RNA chemistry can impact ADAR activity so much. And thanks to the rest of the speakers for giving us so much insight into these novel CRISPR nucleases and what the engineering field has done. The technology that I'm really excited to talk to you about today is all about spatio-temporal control of CRISPR editing and what we've done in Synthego to try to enable these sort of technologies. So you've heard all day about industrialized CRISPR for accelerated discovery to clinic and how Synthego positions ourselves as a company within the field. What I really want to tell you about is kind of this full stack engineering approach that we can bring from a collaboration, pretty much all the way up of these different vertically integrated disciplines, from research to chemistry and bioinformatics, to make a genome engineering product that sits at a sort of intersection between our Halo platform and our research groups. So one thing that we've kind of seen throughout the day is that Cas9 is inherently uncontrollable. Some sgRNAs have high number of off-target sites, such as we've seen over and over again through technologies such as Singdarticide's GuideSeq. And we've also noticed that we have some potential to predict these high-frequency sites, but not always. We can't always rely on the bioinformatics to give us a clear view of the sites that we need to study in further detail. What we've also seen recently is that off-target events can lead to translocation events, and as you can see from this diagram on the left, there are very specific instances such as this MLL-AF9 translocation, which can actually cause leukemias within a mouse model. Really, this boils down to, once introduced, Cas9 RNPs are active until they're degraded by cellular processes, and how I like to analogize this to is sort of a runaway train. We put millions of separate Cas9 ribonucleoproteins into a single cell and then just wait for the cell to turn these over. And that extended amount of activity time can lead to the sort of off-target effects that we want to try to avoid in a clinical type setting. There have been various methods that have been used to try to control CRISPR. Some of the earliest ones split up the Cas9 molecule into the N-terminus and the C-terminus, and with the addition of rapamycin, these can form back together and enter the nucleus to create double-strand breaks. Some fantastic work by my fellow co-panelist Lucas Harrington also has described anti-CRISPR proteins, where it really shows the genetic diversity and the arms race between bacterial and CRISPR about how phages started to evolve and to mutate through these anti-CRISPR proteins to either do active site deactivation or also even completely dimerize with the Cas9 protein and inhibit DNA binding. Other groups have gone with more chemical synthesis moieties, where they've actually photocaged the sgRNAs, where they attach very specific molecules to the uracils within the protospacer of the sgRNA, and with the addition of light, these release, and now the Cas9 is actually able to bind to the target and make a double-strand break. We kind of took the opposite approach to this and said, hey, can we actually control the off rate of the Cas9 RNP system and make a CRISPR sgRNA that fragments in response to light? So in this example, we have what we have called a CRISPR off sgRNA that under normal condition actually behaves exactly as you would expect a normal sgRNA. Has full editing activity, you can prepare and you can handle it the same way. However, with the induction of light, this sgRNA scaffold would actually fall apart and render it completely inactive. Whereas this obviously would not happen with the standard sgRNA. I'll skip you some of the details about all of these sort of proof of concept experiments that we did and some of the further optimization, but what we found is that through the addition of 2 photocleavable moieties in the backbone at very specific positions, we created the system that indeed, with the addition of light, this sgRNA would fall apart, and you could actually, if you see at the graph on the bottom on the left, you could detect each of these individual chemical species. Now, the decision point to put 2 different chemical moieties in it was actually more of a safety switch. We found that by using only a single moiety in there, we were actually able to completely ablate activity, but the fragmentation was not nearly as consistent as we would like. And so by putting 2 in there, we could get a near complete fracture of the CRISPR sgRNA. Again, to make a little bit of a long story short, and you can look at all this data in the Nature Communications paper that we published, but we identified that CRISPR-off is in fact able to modulate double-strand breaks within a population. And if you saw the sort of colors that we had on the previous slide, make sure to pay attention to those. So we have blue, which is our CRISPR-off sgRNA, which has not been exposed to light, green, which has been broken apart, and black and gray are normal sgRNAs. But we saw that there is this huge delta in populations that had been either not exposed or exposed to light, meaning that we could get all or nothing. But we also hypothesized that we could potentially titrate the level of editing events within a population. And that's exactly what we showed in this sort of labor of love experiment, where we harvested each of these different populations 48 hours post-transfection, but actually exposed a different population to light every 2 hours post-transfection, and measured the level of editing activity. And indeed, what we saw and what's really cool about this is that CRISPR-Cas9 editing starts to more look like an enzymatic reaction where you have this very sort of exponential growth before you get saturated editing. And by adjusting the time post-transfection that you actually expose this population to light, you can titrate this level of editing to your desires. We also proved that these CRISPR-off sgRNAs are robust against targets. And so you can see, for example, with your ONPNS1 and your MIP, targets, you're actually getting a very, very significant delta between completely on editing that's very similar to the level that we see with sgRNAs and editing that's completely turned off 4 hours post-transfection. Now, some of you that are eagle-eyed might notice that some of these don't seem to work, such as the FAM163A and the FANC, but actually further experiments on the system showed that this is a really interesting site kinetically, where that the double-strand break and repair actually happens before 4 hours post-transfection, which is when we normally turn it off. So this sort of the kinetics of double-strand break formation and indel repair can happen very, very quickly. And we believe that you can use this system to start to really narrow in and start to focus in on what sort of kinetics are happening over time within an individual population of cells. We also made a very interesting observation that we believe that this system is able to modulate the on to off target editing ratio. And so what you can see is that on the top, there's the on target editing event, which happens very quickly. And then the edits sort of saturate at probably the number of cells that were actually successfully transfected. However, the off target events actually have more of a lag period where it takes them a little bit longer to find those targets and to get going before eventually catching up and having a relatively more saturated editing percentage. We thought that maybe we could expose the CRISPR-off sgRNAs to light at these points to try to maximize the on-to-off targeting ratios. And indeed, that's exactly what we found, where with sgRNAs, didn't matter when we exposed them to light, they had the same sort of on-to-off target editing ratio that we would see at any times. However, if we exposed our CRISPR-off sgRNAs to light either 4, 8, or 16 hours post-transfection, we were still able to obtain very high levels of on-target editing while also mitigating some of the more prevalent off-target edits and completely ablating some of those that are not nearly as common. So far, we've told you about our CRISPR-off system, which is where we take an sgRNA and we're able to break it apart and absolutely shatter it. And how we like to think about this is a little bit like a light switch, where we have a molecule that's completely on, we walk into the room and we turn it off. However, could we also start in the opposite position where we have a molecule that's completely off, we turn it on where and when we want to, and then turn it off all over again? And indeed, we've also managed to create this in a system that we're calling CRISPR-On. Not necessarily the best naming conventions. If you have a better one, please contact me, and we're more than happy to start that discussion. So how do we make this CRISPR-On light switch? And again, to try to, we're not going to go into all the details because it's only a 15-minute talk, but we found that by adding a backbone, with a backtrack, of RNA molecules across that perfectly matches the protospacer, along with the inclusion of 4 different light-sensitive molecules, we were actually able to completely block protospacer binding activity that when we shone light on this entire structure would fall apart and leave a fully functioning sgRNA. And you can see this very nice dose-response curve here at the bottom, where with increasing irradiation time, we have a decrease in the number of inactive molecules as detected by ESI, and we also have a corresponding increase in the number of active sgRNA molecules that are capable of making an edit. We again showed that these CRISPR-On sgRNAs are robust across targets, and so now here we have a CRISPR-On minus light as the green bars, and you can see that there's a much lower level of editing activity, if not completely ablated across a vast majority of these targets. However, when we turn them on, we're actually able to achieve levels of editing that are similar to a normal sgRNA that you would just transfect into your cell and use as typically done. But the pièce de résistance that we want with this is what we're calling CRISPR Lite, which is a fully tunable sgRNA. Now, the majority of experiments that I've talked to you before have this off-the-shelf available PC linker, which is based off of O-nitrobenzyl. But thanks to our really fantastic chemists here at Synthigo, including Anastasia Kanita, we've actually de novo created a coumarin linker, which is based on a completely different photochemistry, but is still capable of linking. The PC linker is broken at 365 nanometers, whereas the coumarin linker is more broken at 420 nanometers. And what that means for us is that we have this really cool bioorthogonal shift where if you look at these optical densities and what kind of energy it's taken to break these sort of bonds, when we expose these cells to 420 nanometer light, we're only going to break the coumarin linker. However, this will not break the PC linker, and we're still going to be capable of using this. Now, combining these, we can create this CRISPR Lite sgRNA, which again is our fully tunable sgRNA. We integrate the coumarin linkers at the places where we want to be able to turn the sgRNA off, and we incorporate the O-nitrobenzyl-based linkers in the backbone where we actually want to be able to turn the CRISPR molecules off. As a proof of principle, we also wanted to show that we could enable orthogonal activation and deactivation of editing. So in this experiment, we transfected a CRISPR-On or CRISPR Lite sgRNA and just let them go. So if you see our normal sgRNAs after 8 hours, we've accumulated a decent amount of edits. However, with our CRISPR Lite sgRNAs, we see 0 level of editing across at this time point. At this time point, we then turned on the molecule and let it accumulate edits for the next 24 hours. Indeed, what you can see is that when the molecule that we turn on has a very similar rate constant to what we're seeing with a normal sgRNA, suggesting that we're able to get the same level of editing that we'd be interested in with a normal sgRNA. More importantly, the inactive molecules are still completely inactive. We don't have any editing at this in this sample. At 24 hours post-transfection, we then turn the molecule off, and what you can see is we're actually getting a separation between the molecules that we left on and the molecules that we then turned off. And the levels of percentage editing does increase a little bit over this time due to indels that are in the cell that are currently still trying to be repaired. We've also shown this at multiple sites, such as EMX1 and STK3, showing that this is more of a generalizable solution for this fully tunable activation and deactivation of editing. So just in conclusion, there's a whole bunch of really other cool applications that we haven't quite told you about, such as this one on the right, where you're actually able to do, with just a simple upright microscope, as well as a thin film mask, you're actually able to do spatial patterning within populations of cells. And of course, we had to draw our logo on it. But really, CRISPR Lite, and what I hope you take away is that CRISPR Lite is a turnkey technology for anyone using Cas9. If you currently work with in vitro transcribed guides, if you currently work with synthetically modified guides, such as what we provide, you can immediately put this into your system and it'll be completely active. It enables a titratable levels of gene editing on a population scale, and that further optimizations allow users to maximize the on to off target editing ratios. We're also continuing to work on this, where future versions are getting longer and longer for both safety as well as penetrance into deeper tissues. We're also really excited about the potential to expand this to other nucleases such as Cas12As, Cas13s, or any of the other many nucleases that you've heard throughout the rest of this session. So with that, I really just want to thank the team at Synthigo. We have a fantastic research team here. You can see we've moved into our brand new building that we're very, very excited about and still building out the labs. And also open up an invitation that we're actively recruiting to work on these projects, to work on other cool, novel projects that are looking at pushing the boundaries of what we can do with CRISPR technologies. So if you're interested in these two, I do have QR codes for both scientists and research associates to work on this. Or if you're really just interested in Synthigo, please visit us at www.synthigo.com/careers. Thanks again to everybody in the audience. This does conclude our World CRISPR Day session on discovery and innovation. Thanks to all the attendees and speakers. And please stay tuned for the upcoming panel with the discussion with the Women of CRISPR, which will start momentarily. You're not going to want to miss it. Thanks again.